Hello, dear students. Welcome back. This is Dr. Anil's Botany. Today, let us see the classification of viruses. How we can classify the virus. There are several classifications are there, but we can discuss some of them one by one. Okay. It's a classical criteria for classification. See, the first classification, no, they are classified according to their symptoms. The viruses shows what type of symptoms. According to their symptoms, we can classify. This is useful for clinical situations. Then second one is host organisms. So the bacteria or the virus which infect which host. For example, in the case of the smallpox viruses infecting the human beings, as well as HBV virus, that means the hepatitis virus B, hepatitis virus. Then physical structure of the virus particle, whether it is enveloped or naked. We have seen in the last class enveloped viruses and naked viruses. Then shape, the helical viruses or icosahedral viruses. All these approaches fail to predict fundamental features of the viruses. Fundamental features of the viruses. The second one is the International Nomenclature Committee suggested 8 criteria for classifying viruses. What are these 8 criteria? The first one is morphology and methods of reproduction. So we have seen the morphology. There may be different shape, structure. Depending upon the morphology and methods of reproduction, whether they are having the lytic or lysogenic type of reproduction. Based on this, we can classify the virus. Then chemical composition and physical properties. Then immunological properties. Susceptibility to physical and chemical agents. The natural methods of transmission, how these viruses are transmitted from one host to the other. Then host tissue and cell tropism. What is tropism? Means the external stimulus, host, tissue and cell tropism. Pathology including inclusion body formation. Symptomatology. These are the 8 criteria for the International Nomenclature Committee suggested to classify the virus. So modern criteria for classification, the first thing is the classical way. The viruses infecting animal or plant or bacterial, that type of, that is the classical type, whether it is enveloped or naked. Then second thing is genomic, whether it is DNA or RNA. That was proposed by Baltimore. Baltimore scientists classified these viruses into, according to their genomic structure. Then the serology, that is diagnostic virology, classification based on diagnostic way. For example, in the case of infectious bronchitis virus, IBV of chickens, that is a type of coronavirus that now we have facing this disease, coronavirus. In this coronavirus infecting chickens, there are 3 different types present. And all these types, have significant antigenic differences. Antigenic differences are there, but perhaps very little genomic or biological difference between these viruses. There are 3 types, and these 3 types have antigenic differences are there, but very little genetic or the biological difference between these viruses. So likewise, we can classify the viruses. The another classification proposed by Holmes, and he formulated the Carolus Linnaeus binomial nomenclature system. So Carl Linnaeus, you might have studied Carl Linnaeus classification. That is one of the sexual system of classification proposed in the case of angiosperm taxonomy. Naming of plants, that's called Linnaeus classification. and he classified virus into 3 groups under one order, that is virales. Holmes classified the entire group of virus, that is order virales. That the whenever you classify the order, then you can end with a suffix ales, that is order virales. And this order virales is classified into 3 groups, and the first group is the phaginae, that means it attacks bacteria. And the second group is phytophaginae. Phyto means something related with the plants, so it is called the virus which attacks plants, phytophaginae. And the zoophaginae, that attacks animals. So likewise the Holmes classified. And these 3 scientists, Andrew Loff and W. Horn and P. Turnier, these 3 scientists, I have collected only 2 photographs of these famous scientists. I didn't get the Horn's photograph, and fortunately we have got to Andrew Loff and Turnier the photographs. You just see the famous scientist. That is why this classification is called LST, system of virus classification. LST system of the viral classification. So in this way we can classify the viruses. Shape, then cell structure, then the combination of antigenic properties, or the different types of chemical combinations present in the outer surface of the body of virus. Likewise, the scientists try to classify the viruses. But the most advanced type was proposed by the LSG system of classification. That is, main characteristics used are nature of the nucleic acid, whether the nucleic acid is RNA or DNA, then symmetry of the capsid, presence or absence of an envelope, that is enveloped or naked viruses, dimensions of the virion and capsid. Virion is a complete infectious particle, is called virion, and the capsid, the number of the capsomeres. And this is the most accepted type of classification that was approved by the Professional Committee on Nomenclature of Viruses, that is called PNVC, the International Association of Microbiological Societies, that was proposed in 1962. So that is the phylum Vira is divided into 2 subphylums, Deoxyvira and Subphylum Ribeira, right? Then what is Deoxyvira? The DNA viruses. The viruses have deoxyribonucleic acid is the genetic material, and the Ribovira means the genetic material is RNA, ribonucleic acid. So deoxyribonucleic acid and ribonucleic acid, that 2 subphylums are classified. Then subphylum Deoxyriba, Deoxyvira, that's a subphylum Deoxyvira. DNA viruses are classified into or grouped into 3 classes, that is class Deoxybinala, class Deoxyhelica, class Deoxycubica. So depending upon the the symmetry, we have seen the helical symmetry, we have seen the icosahedral symmetry, we have seen the cubical symmetry. Depending upon the symmetry, the deoxyriba is divided into 3 classes, that is class Deoxybinala, that means bi means 2, dual symmetry. Then class Deoxyhelica, helical symmetry, cubica means cubical symmetry. Then these classes are divided into orders. The class Deoxybinala is divided into order Urovirales. Deoxyhelica is divided into order Chytovirales. Then class Deoxycubica is divided into 2 orders, that is order Peplovirales and order Haplovirales. So whenever I told you earlier, then classified into order, then order must be end in a suffix alias. That is why the class is divided into order. Then order have urovirales, cytovirales, papillovirales, and haplovirales. Haplovirales means no envelope. They are naked viruses. See, then order, then coming to family. So each order having one or more families. Then order Eurovirales have only one family, that is Phagoviridae. Then family end in Viridae. Not like in the case of Angiosperms, the family is ending in ACE suffix, that is Papillonaceae, Cisalpinaceae, Rubiaceae, Malvaceae, right? So likewise, in the case of angiosperm classifications family, that end with and suffix ACAE. But here the family end with Viridae, order ALES suffix. See, a family Phagoviridae. Then order Cytovirales have only one family, that is Poxviridae. Order Papillovirales have only one family, that is Herpes viridae, have 162 capsomeres. They have cubical symmetry. And order Haplovirales, without any envelope, no envelope, without envelope, we can say naked, that Haplovirales have 5 family. That is family Iridoviridae, have 812 capsomeres. The number of capsomeres is also taken into account for classifying this virus. That is family Iridoviridae have 812 capsomeres. Then family Adenoviridae have 252 capsomeres. So the capsomeres, small subunits called monomeres, and small spherical bodies, small the beetle-like structures that united together. That is 812 capsomeres, 252 capsomeres. Family in the case of Papilloviridae have 72 capsomeres, and in the case of Paroviridae have 32 capsomeres, and in the case of family Microviridae have 12 capsomeres. Could you follow me? So the subphylum Deoxyvira is divided into 3 classes: class Deoxybinala, class Deoxyhelica, class Deoxycubica. So class Deoxybinala means dual bi means 2, dual symmetry. Helica means helical symmetry, cubica means cubical symmetry. So likewise they classify. Then class is divided into order. Order Euroviridales. In the case of Deoxybinala have only one order, that is Eurovirales. Eurovirales have only one family, that is Phagoviridae. Then class Deoxyhelica have only one order, that is Cytovirales. Cytovirales have only one family, that is Poxviridae. But in the case of Deoxycubica have 2 orders, order Papillovirales and order Haplovirales. And in the case of order Papillovirales have only one family, Herpes viridae. But order Haplovirales have 5 families. See, that is Iridoviridae, Adenoviridae, Papilloviridae, Paroviridae, and Microviridae. That much about the Deoxyvira. Then coming to Ribovira. The Subphylum Ribeira have 2 classes. That is class Ribocubica and class Ribohelica. Ribocubica and Ribohelica. The Ribocubica have 2 orders. That is order Togovirales and order Limovirales. Togovirales and Limovirales. Togovirales have only one family, that is Arboviridae. But in the case of Limovirales have 2 families, that is family Napoviridae and family Rioviridae. But in the case of Ribohelica, helical symmetry, and Ribohelica have 2 orders, Sagovirales and Rabidovirales. Sagovirales and Rabidovirales. And these Sagovirales have 3 families, Stomatoviridae, Ramixoviridae, and Mixoviridae. That is three families. Sagovirales have three families: Tomatoviridae, Ramixoviridae, and Myxoviridae. And order Rabidovirales have two suborders. That is, order is again divided into two suborders: suborder Flexivirales and suborder Rigidovirales. Flexi and rigid. That is why it is called Flexivirales and Rigidovirales. And Flexivirales have a two family, that is Mesoviridae and Peptoviridae. Mesoviridae and Peptoviridae. Suborder Flexivirales have two families, and Mesoviridae and Peptoviridae. Suborder Rigidovirales have three families: Pachyviridae, Protoviridae, and the Polycoviridae. So the Subphylum Ribovira have two classes: class Ribocubica and class Ribohelica. Could you follow me? Just like in the case of Subphylum Deoxyvira, there we have three classes, right? Three classes. What are they? That is Deoxybinala, Deoxyhelica, and Deoxycubica. But in the case of Ribovira, there you can see two classes, that class Ribocubica and Ribohelica, right? And this ribocubica is divided into two orders, that is togovirales and limovirales. And these togovirales have only one family, that is arboviridae. But limovirales have two families, napoviridae and rioviridae. Could you follow me? Right? In the case of Ribohelica, have two orders, Sagovirales and Rabidovirales. Sagovirales have three families, Stomatoviridae, Ramixoviridae, and the Myxoviridae. But in the case of Rabidovirales, that order is again divided into two suborders. The suborder Flexivirales and suborder Rigidovirales. One form is flexible and another one is very rigid. That is why Flexivirales and Rigidovirales. And in these Flexivirales have two families, the family Mesoviridae and Peptoviridae. But in the case of Rigidovirales have three families, Pachyviridae, Protoviridae, and Polycoviridae. So that is the the classification of viruses. Any doubt in this? It is somewhat difficult to understand all these terms and other things. Once you go through this, you can easily catch the deoxyribovirus ribovirus and ribonuclear DNA viruses and RNA viruses. Likewise, that is they have classified, that is LST, system of classification. That is very easy to follow. Okay, could you follow me? Any difficult you have faced, you tell me directly, we can simplify as much as possible. So let's move on to another classification, that is Casjens and Kings classification of viruses. The Casjens and Kings classification of viruses. Casjens and Kings 1975 classified virus into four groups based on nucleic acid, presence of envelope, symmetry, and the site of assembly. The Casjens and Kings classification. The types of nucleic acid, envelope, symmetry, and assembly. So Casjens and Kings classification of viruses, it is mainly based on the genetic material present in them. Maybe RNA viruses or DNA viruses. RNA may be single-stranded RNA virus or double-stranded RNA virus. Single-stranded DNA virus or double-stranded DNA virus. Likewise, he classified. Another classification proposed by International Committee on Taxonomy of Viruses, that is ICTV classification, International Committee on Taxonomy, International Committee on Taxonomy of Viruses, that is ICTV classification, established in 1966. So that body takes the task of developing, refining, and maintaining the universal virus taxonomy, that is classification and nomenclature also. governed by the Virology Division of the International Union of Microbiological Society. See, this ICTV classification, the viral classification starts at the level of order and continues as follows. You can see the order virales. I told you earlier, whenever you classify the order level, then the order should be end with a suffix ALES, order virales. Family Viridae. It's not like in the case of angiosperm taxonomy. There the family end with the ACAE suffix. But here, no, it is Viridae. Then subfamily Virinae, right? Then genus, virus, and species name is according to the symptom or the disease. Likewise, the ICTV classified the viruses or virales, viridae, virinae. That means order, family, subfamily, genus, and species. This is the hierarchical system of classification. So that much about the classification of viruses. Different types of classification we have seen. Then the nomenclature of viruses. How we can name a particular viruses. There's various approaches are there. That do not obey the binomial nomenclature. But the binomial nomenclature is, the scientific name of each and every organism have two sections, that is genus name and species name. Genus name always starts with a capital letter and species name always starts with a small letter. And after that, you can write the author's name who invented that particular species. Likewise, you can name. But in this virus classification, that do not obey the binomial nomenclature. And the first thing is, they have named after the disease. What type of disease they produce? Measles virus. The virus which cause measles. The virus which causes smallpox. Likewise, measles virus, smallpox viruses, named after diseases. Then name after the places where the disease first reported. The Newcastle disease virus, the Ebola virus, Norwalk virus. The places in which the particular disease originated. Then coming to the host and signs of diseases, symptoms of diseases, and the host. For example, in the case of tobacco mosaic virus, the tobacco plant shows mosaic pattern. So it is called tobacco mosaic virus. Then cauliflower mosaic virus, in the case of cauliflower, and which shows mosaic symptoms. That is why it is called cauliflower mosaic virus. Brew mosaic virus. See, then according to the the languages, Latin and Greek words, using the viruses, corona viridae, that means crown. Corona viridae means crown. Then Latin and Greek words. Then parvo viridae means small viruses. Then coming to virus discoverers, who discover first. For example, Epstein-Barr virus. Then how they were originally thought to be contracted. For example, dengue virus, evil spirit, influenza virus. The influence means bad air. Likewise, they named. Then the combinations of the rose sarcoma viruses. And that likewise we can name the viruses. So that much about viruses. Could you follow me? This much about the classification and nomenclature of viruses. It's almost a dry area, but we should understand how we can know the classify and name meaning of viruses. Okay, any doubt in this? Thank you very much. Thank you for patience.